then it's probably not heritable and we can just go home. So now that you guys know how to read one of these tables, don't embarrass me now. All right, so did we get a significant effect of strain? Why do you tell me these things? What's less than 0.05? This one? This one? Which one? That one? Strain. All right. So we got a significant effect of strain. So that suggests that indeed, yes, our residuals are different, that the genotype is going to matter, and we actually also, out of this analysis, got a little bit of a heritability estimate. So about 0.41-ish, okay, was our heritability estimate. And that's good news because we need at least 10% for heritability for the QTL to work. So that's what we got out of that. Okay, one of my other goals for this unit was to introduce QTL analysis. And I'm not kidding you when I tell you just about every talk I go to anymore has a QTL analysis in it. Since I last lectured to you, I went to a talk and they were doing QTLs on chronic fatigue syndrome. Another guy was doing QTLs on pain sensitivity. So just about anything that has a genetic basis, people are now doing this QTL analysis, trying to figure out what aspects of chromosomes may be contributing to a phenotype. And if you know that, then you can zoom in and figure out what genes in that hunk of chromosome might be contributing. So there's a lot of this going on. It's a technique that you're going to be seeing probably in the rest of your life, and it's getting more and more refined. So I wanted you to know about it. QTL analysis, as I said, is good for detecting the approximate locus of multiple genes affecting the phenotype. We got, I think, 3 peaks, right, in our data, 2 on 6 and 1 on 2. And so that's across all the finding the locus of multiple genes affecting the phenotype across the whole genotype, except not Y. And I have asked you before, and I ask you now again, why not? Y doesn't recombine, so you can't really use this analysis to look at the Y chromosome. Okay. We've already talked about the fact that we were using recombinant inbred strains and that there were markers on, and so what somebody has done to emphasize this again is for every last one of those thousands of markers across the whole genome, somebody has done the PCR reaction and figured out, does that recombinant inbred strain have the B marker or the D marker? So there's a lot of molecular biologists with sore thumbs who've already done this work for us. It's already in the databases, so we can use it. Already been done. So we already know, does that recombinant inbred strain have the B form or the D form? Again, it uses the likelihood ratio statistic, and now let's talk a little bit about what the likelihood ratio statistic does again. And what the likelihood ratio statistic does is, first of all, at a given locus or marker, it takes all of the animals that have the B form and puts them in one pile, and all that have the D form of the DNA puts them in the other pile. All right? Then what it does is looks at the phenotype, or in our case, the residual. So back in the numerator of this thing, it looks at every value minus the mean of all and squares them up. So what it's going to do is take every value minus the mean and then square that value. That's the numerator. Okay? And then the denominator, it divides it up with regard to groups. So it says, okay, all those in the B group, we're going to take each value minus the mean of the B's and square that. And all those in the D group, we're going to take each of those values minus the mean of the D's and square that. So if the data looked like that, in this particular example, and we had this thing to grind through, would this value end up being large or small? Large. Okay, so my point is, when you get the data to look like this, that everybody who's got the B form of the marker, their phenotype is highly discrepant from those that have the D form of the marker, that's when the likelihood ratio statistic is high. That's when we'll be getting those peaks in our QTL analysis. And that's when, and what that suggests is around that marker, or even at that marker, there are genes that might have had a big impact on that phenotype. In contrast, if it goes in, let's say it goes to the next marker and separates all those that happen to have the B form from all those that happen to have the D form, and you get this kind of a spread in the residual. So you can see that everybody here with the B form, the variance away from the grand mean, the mean of everybody, and the mean of them is pretty much the same, and the variance of everybody around the grand mean and the mean of this marker is going to be pretty much the same. Is your statistic then going to be high or low? Low. Okay, so here you can see just at a glance the phenotype is not highly discrepant. Okay, and so then we'll get a place where the likelihood ratio statistic is low. And would you expect there to be genes around this marker that would influence the phenotype? No. All right. So that's what QTL analysis does. And again, this is not your data. It's pretty close to your data. We typically, when we've done this, have gotten a peak on chromosome 6. I think you guys found 2 little peaks on chromosome 6. So again, what it's doing is it's finding the likelihood ratio statistic as a function of all those different markers, lots of different markers across the whole genotype, and doing that likelihood ratio statistic at every last one of those markers. So this blue line here represents the likelihood ratio statistic as it's changing as a function of the markers that are on, and these numbers represent the chromosomes laid end to end to end to end to end to end to end. And by the way, I don't think I've mentioned, but it does pop up in your reading, there's a standard way to array the chromosomes, and the left-hand side is always called the proximal, and the right-hand side is always called the distal. So in your readings, that Williams et al., they'll talk about things being on the proximal or distal end of chromosome 6. That means on the left-hand or right-hand way, but there's a standard way that chromosomes are laid out when people do this and talk about it. Okay? Okay, so squiggly blue line is the likelihood ratio statistic, and this line here is the suggested line, and this line up there is the criteria, is the significant line. Again, we only got things that were suggested, but at this juncture, as I speak to you today, QTL is basically a fishing expedition, so I don't feel real bad as long as we got something that's in the suggested range. If I were in a real lab, I'd go and look under those peaks and see what was there. I wouldn't just say, oh, wasted my time. I'd go look, and I'm sure people do. Actually, I've seen things reported in the literature. Matter of fact, I think it was your paper, Rick, where you did talk about a suggested marker, right? Of course. Yes. All right. This, by the way, isn't your data, but it looks pretty similar. Again, we zoomed in on chromosome 6, and we talked about the fact that we wanted to go look in especially places where these lines here represent the SNP density. The more lines, the more SNPs. Would you want to go and look in places where the SNP density is high or low? Why? You got to yell at me. I'm half deaf. Single nucleotide polymorphism. Why would you like lots of those? Because there's variation in the genes. Variation in the genes might translate into variations in the phenotype, right? If everybody's got the same gene, is it likely to have a differential phenotype in its expression? Yeah, probably not. All right, so we looked where the SNP density was high, and we also then bounced ourselves into the UC Santa Cruz genome browser from this, and I'll be talking about that ere long. Okay. And in fact, right now. So the other thing I wanted you guys to see was a whole bunch of bioinformatic tools, and that was one of the focus of this module. These are tools that even as I speak, real deal scientists, biologists are using right now to figure out questions like we address. You know, what's affecting a phenotype? How are genes different between a disease state and a non-disease state? Things like this. So these tools are out there. You may recall when I first started this unit, I talked about Gregor Mendel and how his paper essentially got lost for 40 years. People forgot about it. We're generating piles of data right now. We want to keep access to it, and some far-sighted people have created these bioinformatics